Okay. Hello, everyone. Thank you for coming. And I'm very excited to present Dr. Elizabeth Clark. I met Lise back in 2022 when I was starting to learn all about CT scanning. She has been one of my micro CT and synchrotron gurus in my path of learning 3D imaging. She has a background in both biology and paleontology, working with a variety of animals, from marine invertebrates to land insects and all kinds of critters and cool stuff. And now she's working as a beam scientist at the Berkeley Advanced Light Source Facility at the Berkeley Lawrence National Laboratory. And I'm not going to jump too much into the details of all her work because she's going to be sharing many of those today. So with that, please thank you for joining us. And all right.
Hi, I'm Liz. As Denise said, I'm a scientist at Lawrence Berkeley National Laboratory, which is the national lab in Berkeley. And I'm a biologist studying organismal biomechanics and form and function. Most people, I think, maybe not all of us, but maybe your friends and family, when they think about what an entomologist or any type of organismal biologist does every day, probably looks something like this, right? We run around with nets and have like bugs crawling on our heads, like, and some days, probably like all of you, that is what I do. I do like going out in the field and collecting and examining bugs and plants and fossils in their natural habitat, running around with a net. But more often, my day usually looks like this. I'm a Beamline scientist at the ALS, the Advanced Light Source, which is a particle accelerator in Berkeley. And in my research, I use 3D imaging from the X-rays that are generated by the particle accelerator to digitally visualize 3D structure in organisms, to look at the relationship between structure and function, to answer some fundamental questions about the inner workings of animals and plants and all kinds of creatures. So I've worked on a lot of different groups. As Den said, I did my PhD on echinoderms, where I was looking at the origin and evolution of certain strategies for movement and locomotion. And then I did a postdoc in paleobotany. Since plants do everything that animals have to do, but animals use motion to get around and make it happen, and plants have to stand still. So I got pretty interested in some of the structures that plants use to make their lives happen. And most recently, I did a postdoc with Rodrigo Almeida, if some of you know him, at UC Berkeley, where I started becoming really obsessed with insects. In terms of interesting structure and function, I think this group really takes the cake. Because they do so many things, amazing things, better than all other animals put together. I hope that's not too controversial. They can eat more than anybody else. They're faster, they're stronger, there's so many of them, and so many different types of them all over the world in all kinds of different environments. And they have really specialized tools for taking in information, for learning, for communicating, for navigating, and for structuring and shaping their environments, which makes them a really exciting group as somebody who's interested in structure and function. So I've kind of gone down this rabbit hole for the past few years, and I think there's some runway there ahead. So today, I have a few agenda items I'd like to accomplish here with this talk. It's a crazy world we live in, so if anything, just sit back and relax and maybe learn a couple cool things about bugs. So also, a lot of people don't like bugs. I imagine most people like bugs here, I would imagine, right? Like we got a lot of, yeah, this is a very bug-friendly audience. I think that was 100% of hands. So I do appreciate that. So you can leave if you so once we're kind of, that goal, it seems like it's already been accomplished. But then my next item is that I want to show how 3D imaging can help us understand them better. It's a really exciting tool. I know it's something that's been around for a while. I'm sure you have all been to talks about how 3D imaging can be used to study organisms in one way or another by this point. But there's a lot of really emerging and exciting developments with the tool that's always kind of growing and changing with new technology, new approaches for studying things. So I hope that's something I'm excited to convey. And to do that, I'm going to talk about an extreme insect behavior that I study, that I've used the ALS to learn more about. And then I'm going to talk about some of my hopes and dreams, share those with you. So I'm going to get into our work on feeding first and share a story of an extreme insect behavior to demonstrate how we use some tools at the ALS to dive deeper into the biomechanics of how all of these extreme behaviors work. So does anybody know what bug this is? Yeah, oh, I love it. Yes, this is anybody down to the species level? Oh, that would have blown me away. This is a blue-green sharpshooter. So as all of you know, it lives here in California, and it's part of a group, as you already know, I'm sure, called xylem sap feeding insects. So what's exciting and interesting about them, for those that aren't familiar, is that they have these little tiny needle-like mouth parts that they insert into the plant that they use to extract the xylem sap. And that's interesting for a couple different reasons. I'm sure you know that xylem sap is really low in like its nutritional value. So they end up having to feed somewhere between 300 to 1,000 times their body mass every day. So I looked up what 1,000 times my body mass is, and I found a community food drive that made the news. So imagine having to eat this every single day just to meet your metabolic needs. And for those of you who are interested in like the physics of xylem sap, like I know some of you here are, like my friend Andrew, who that's his primary area of research. Xylem sap, the tension within xylem sap can be under about or more than -2 megapascal. I'm suddenly nervous because I don't want to get something wrong. But it can exceed -2 megapascals of tension. So there's some really interesting questions about how these small insects are able to overcome the tension in the xylem vessels to extract sap. I kind of imagine like you're trying to get your metabolic needs by like sucking on a piece of celery or something like that. It's like there's a lot of tension you have to overcome. So you can imagine, if you don't care about physics, you can imagine you have to fight all of the volunteers in this room in order to get the food out. Like every bite, you have to like push people away that like really want to hold on to the food. So yeah, these insects are a bit of a conundrum, which as a biomechanist, as an organismal biomechanist, I find very fascinating. And on top of that, as I'm sure many of you are familiar with, these xylem sap feeders, like our beautiful blue-green sharpshooter, can bypass a plant's defenses when they feed with those little darn needle faces of theirs, and can pass pathogens in the process of feeding, like Pierce's disease to olives and grapevines, which are important for California and global agricultural economy. And right now there's not a cure, so the kind of aim of this work was to have a better understanding of how these insects are able to feed in order to kind of understand maybe some strategies for preventing the transmission of Pierce's disease, or at least understand how it's happening in the process. But the problem with that is that we don't really understand the mechanics of how they're doing all of this. And that's because all of this is happening inside the plant, right? I kind of liken it to if you were a scientist and you are at In-N-Out and you want to study people eating, it's a lot easier to study somebody eating a hamburger than drinking a milkshake, right? Because you can like see the bites people are taking, you can see like how many hamburgers they're eating versus a milkshake. It's like, it's all kind of happening inside. So it's difficult to kind of figure out like the rate of consumption or the volume and that kind of thing. So that's how 3D imaging comes into this. 3D imaging as a scientist is really my bread and butter. I've worked on a lot of different groups, but I've used 3D imaging as a tool to ask different kinds of questions about all of them, because there's a lot of like really exciting things that we can learn that we can't really learn any other way. Like a dissection, it's a great strategy for looking at small structures, but using 3D imaging, you don't have to cut things apart. You don't have to break apart to see things on the inside. You can see articulated anatomy really nicely, as well as internal structures in 3D. And once we have the 3D imaging data, we can export it to do a lot of really interesting computational methods. Does anybody, I imagine some of you probably use 3D imaging here too. So I'll be excited to hear what you're working on. And for those of you who haven't, it would be exciting to kind of get the wheels turning about like what you could do using this kind of technique. So the way it works, I'm not going to get super into the weeds, but it's a pretty straightforward concept. This is a X-ray of my spouse who's turning 30 this month. But this is an image for when they were 7 years old and they swallowed 2 quarters. And I not only use this in every talk I give to kind of like razz them a little bit, but also because it's a really great way to demonstrate how X-rays work. Like we're all familiar with like maybe seeing a skeleton or a bone, but having the quarters really shows the way that the X-rays interact with things of different densities, right? So the quarters are super dense. So when the X-rays hit the quarters, they get absorbed and leave kind of like a shadow on the image screen behind them. When they go through your bones, they do absorb some, but not as much as something super dense like a quarter. And then your soft tissue, the X-rays just kind of float through. So it's basically exactly the same. We, at the ALS, the accelerator generates some X-rays and we have, or sorry, I'll take a step back. Just to do a 3D image, you have X-rays that are hitting something exactly the same way at the doctor or the dentist. But that thing is spinning around. So you get 360-degree X-ray projections. And then a computer can stitch those all together to create a 3D image of the object based on the density of the different material in the X-ray. So a lot of you are probably familiar with a medical scanner, hopefully not too familiar. But basically the X-ray source and the detector like spin around your head, which is kind of cool. They even have, I mean, maybe at Davis, do you have one of these? Yes. You do? So a horse CT scanner. So I know people who study dinosaurs or whale bones or whatever and use like an agricultural animal horse CT. I would love to check that out someday. Maybe I could swing by there sometime. I think that would be really fun. But then there's also micro CT scanners. There's one here as well. If you want to scan something the size of a baseball. But in order to scan things that are smaller than that, something the size of a seed maybe, or a bug, for instance, we really need a concentrated stream of X-ray particles to get enough to really resolve the density differences in the small structures. So we use particle accelerator for that. And basically how this works is that there is a linear accelerator in the middle, just like, I don't know, CERN, but they're firing things at each other. And in this existence, we're all kind of moving in the same direction. So the particles go into this ring here and they get sped up to about 99.99999.5% the speed of light. And then they get shot into this accumulator, the storage ring here, as we call it. And in order to get them to move in a circle, they don't really want to go in a circle. So there's a lot of magnets that turn them a little tiny bit. And every time, this is the best explanation I've heard from it, from people that work there. Maybe I need to talk to different people, but the electrons get mad that they're getting bent by the magnets and they spew off X-rays. So at every juncture with the magnet, we just get a ton of X-rays that are getting sprayed off. And at my station where I work, we do 3D imaging with the X-rays. So in this huge building, we end up scanning teeny tiny little specimens. My Beamline is actually one of the bigger things that we look at. Most people are looking at like atomic spin of an atom or whatever. I don't know. I didn't say that. Don't tell anybody. But stuff that's smaller, physics stuff that's a little bit over my head. I just do the bugs, okay? So here's a little tiny sharpshooter in our experiment station here, which is already like the size of a room. So we're doing something even smaller. And here's the schematic that I showed before. We have the X-ray source coming through that little orange box there. The X-rays go, hit the object, and we have a tiny little detector about the size of a quarter, don't tell my partner, that picks up the X-rays. So getting back to our question about xylem sap feeders, how do we use 3D imaging to understand this? So first, obviously, we do 3D imaging. So we do this to get a really nice high resolution image of our sharpshooters that shows us the anatomy of the different parts inside the head that are important for feeding. So this here, if I move around, is that bad for the zoom, by the way? Okay. So this is a pump called a cibarium that opens and closes. They stick their little needle into the plant, and then they open and close this pump to pull sap up the stylet and then into their esophagus. And this giant muscle here that takes up like most of their head is what's powering the pump to open and close. It's a super powerful muscle, which is really cool. And using the synchrotron, we can see nuances in the anatomy that you can't really see any other way, down to a few microns, like this teeny tiny valve that opens and closes. We think it has to do with like regulating the pressure as the insect like pulls its stylet out of the, or maybe inserts the stylet into the plant, that kind of thing. We don't really know, so that's something that we're looking at. This muscle used to have a different name, but after I got this scan, I called my postdoc advisor and I was like, hey, why is the muscle called the, it was called the BLI muscle? I was like, why is it called the BLI muscle if it's not connected to the BLI? And he was like, what? So that's just, it's a small nuance, but it was exciting to like realize that this isn't, that it had a slightly different function from what we thought it did before. That kind of changes a little bit of our interpretation of like how the fluid mechanics work. Then pulling back to the bigger picture outside the nuances of the anatomy of like one insect, we know that these insects have differential like rates of feeding, rates of transmission of Xylella fastidiosa, the bacteria that causes Pierce's disease. So we can ask some questions about like how their anatomy and the differences in their anatomy and the sizes of their muscles, the power of their muscles can influence some of these features as well. And the first step we kind of took to answering those types of questions was by building a computational fluid dynamics model using the anatomical measurements that we took from the 3D imaging data to look at what's going on inside the plant at the plant and insect interface. So I'll just talk about that really briefly. So there's a lot of like nuance and intricacy, and it's pretty computationally intensive to build a model. So we built a very, very simplified version of like this interface between the food canal and the xylem conduit, just to get down to like the very basics of like how the system works, and also to play around with some parameters like, I don't know, the width of the xylem, right? Because a lot of plants have different xylem widths. So like, how does that influence how the insects have to approach feeding? And we worked with Brent Gibson at UT Martin, faculty member who we worked on the fluid modeling together. Andrew Macaron and Craig Broderson. Andrew's here. Sorry I keep talking about you. I don't know if that's making you uncomfortable, but Andrew's here again. He hasn't left yet. So he and Craig do really interesting and amazing work on xylem fluid dynamics using 3D imaging as well. I think you guys were the first people to scan a plant to do 3D imaging with plants. So like they thought it couldn't be done, and they were like, okay, we're just gonna put a plant in a scanner now and like see. And it worked, and it's amazing. And there's like so much intricacy like within and between the xylem vessels inside the plant and the other parts of the anatomy. And they like kind of together realized it's so much more complex and nuanced that like then like these kind of histological slices that you get kind of in your biology textbook, which is really exciting. So we worked with that. I did my postdoc with them, so we were able to kind of integrate like these two, like our insect work and our plants work together to kind of examine this interface from like a really basic fundamental level. I'm almost done with the reviews and we're going to resubmit. So again, don't worry. Don't yell at me. So we answered a couple different kinds of questions using this model, just about like the basic fundamentals of what's going on. And one of the questions we asked, for example, was how much force do insects need to meet their daily needs? So using our modeling, we could perform some simulations, asking these questions and comparing different taxa. So we varied the stylet pressure and looked at how that changed the rate that fluid was moving through the little tube of the bug. So for example, we looked at the blue-green sharpshooter, which had a 13 micron width of the feeding complex. And we looked at how that would influence, so stylet pressure at 13 microns would change the velocity magnitude, and then we compared that to what's actually been observed in terms of ingestion velocity. And this is kind of like a pretty, like a metric that's based on like excretion values, and that can kind of change. So it's not necessarily like the best to lean on, but it's kind of the best that we have. So just from this, it was kind of interesting to learn that these guys only need to exceed the pressure in the xylem vessels by about less, like 1% or less, in order to meet their daily needs. So even though they're drinking so much from the plant, they only need to exert a little bit more pressure than what's in the xylem vessel in order to make that happen, which is kind of cool. So we also looked at a couple other things, like I wanted to see like what, if you were a sharpshooter, could you kind of set yourself up for success? Like, could you pick a particular xylem vessel, for example, or like insert your stylet at a certain angle and that kind of thing? And we found that if they pick a wider vessel at an acute angle, it's not, I'm going to take out acute angle because it wasn't, we reran the analyses with it's within the error range, so kind of ignore that. But if any of you go out xylem sap feeding later, it might not be that big of a deal. At a relatively shallow depth of penetration into the xylem vessel. So yeah, we were able to kind of use our model to look at a couple, gain some insights on how insects might be able to, are actually able to accomplish this amazing feat using the parameters that we've driven from our 3D imaging data, which was really exciting. So even though these are great insights, at the end of the day, we're restricted to looking at static images of insects post-mortem, right? But my dream has always been to examine 3D structure and function of insects engaged in real behavior, to be able to calculate like fluid flowing through the insect as it's happening. So we don't have to rely on these like measurements that are from excretion over the course of the day that have a lot of error, that don't necessarily reflect what's going on at the feeding end. Are any of you familiar with XROM? Okay, it's okay, yeah. It's basically a technique where, I should have had a slide about this, where people put 2 X-rays and project it onto an animal doing a behavior. I did it with like frog, or I didn't do it. I just observed people doing it with frogs one time, where you're like taking 2 X-ray videos of a frog jumping, and then you CT scan, it has little markers embedded, and then you CT scan the frog, and you get this like 3D picture of the frog while it's jumping. And because you have the X-ray videos and the CT data, you're able to like piece them together to get some like cool biomechanical data out of it. That has always been my dream, but the problem is the bugs are so small, you can't embed markers. And also we can't have 2 different X-ray sources because we just have like the beam, and it's like, you can't like really shift where the X-rays are coming from. So we kind of like pieced, thanks to Andrew's help and some other folks that we work with, we were able to kind of like piecemeal a method together that allowed us to do that, which I'm really excited about. So the first thing we did was live X-ray imaging. And the second thing we did was 3D imaging of flash frozen specimens, so we could get like snapshots of what they're doing. So this is this is our bug here. This is the stylet inserted into the xylem vessel, and it's opening and closing the cibarium here. So how that's working is we brought the bugs to the synchrotron in Chicago, and it's the same thing as like what happens at my Beamline, but the bug isn't spinning around. So we're getting an X-ray video of the bug feeding while the X-rays are kind of being shot at it at about 50 frames per second. And this was a technique invented by Jake and Kamel at Argonne National Labs that they were using to study insect respiration. And we wanted to see if we could do it with the bugs feeding. So we just kind of brought a bunch of basil plants and some bugs to Chicago. And but at the end of the day, there's only so much that you can get from 2D alone. Like we can look at the rate of cibarial contraction, but that's, and maybe make some volume estimates, but that's kind of it for now. So we wanted to see what the cibarium was like in 3D during feeding so that we could calculate the volume of a given pump and look at the rate and periodicity of fluid consumption over time by coupling that with our 2D video data. So one of my now friends, Harold at the Beamline, created this tool that we call the CryoJet. And it's for imaging frozen specimens. So basically we flash froze insects during different stages of feeding. And we kept this on. It was like spraying super cold liquid nitrogen vapor over the bugs so that they would maintain a frozen temperature while we were doing the 3D image. So we could keep that snapshot intact and visualize it in 3D. So we're able to get images of the cibarium when it was empty, when it was full, and when it was basically like doing the equivalent of swallowing. I don't know if that's what they call it in bugs, but basically swallowing the xylem sap. So we can look at, and then by coupling these together, like we basically can use the 3D data that we collected. There's a relationship between if you take a picture of it in 2D and measure the height, you can back calculate the volume of the 3D structure. We found a really tight relationship there. So we can take that and we can go back into our 2D imaging data and calculate the volume of every single pump that we saw in the videos. So there's like hundreds, we have like 500 of these or something like that. So we're able to derive like the rate of consumption, the periodicity, how that can vary over time, which give us this really nice comprehensive look at what's going on in the head of the insect during feeding. And I think that has some interesting implications when we if we were to go back and like integrate this into the fluid dynamics model that we have, we could, or even like look at something localized, like that little flap that I showed is like pretty important for, basically found that like if the insect has bacteria there, it's correlated with a plant becoming infected with the pathogen. So there's some like interesting things that we could kind of do in there. If anybody wants to do a postdoc with me about that. But it also, I think, is a really exciting and interesting opportunity to think about like what other kinds of questions we could ask using this like couple technique with other types of insects that are doing like really cool and interesting things. A quick aside, in order to do this 2D X-ray video component, we had to fly the sharpshooters to Chicago in our carry-on on the airplane, which was a real pain in the ass. We obviously had to get like permits to do this. We had to call the airport, TSA, the airline, like United couldn't understand why, like that this wasn't a pet. They were like, oh, it's only dogs and cats are the only pets allowed. I'm like, they're not really pets. It was a very kind of like interesting experience. So I just wanted to throw in, and I was at my cousin's wedding, so my friend and collaborator Andrea had to take them through the airport for me. So I just always want to show my appreciation to her because it was kind of a pain. And we also had to build them a little winter jacket because it was winter. It was November in Chicago, so there was a lot of things to kind of consider. And we couldn't leave them without food because they're eating 1,000 times their body mass every day. So we had to like grow these little tiny plants and like put them in these like little nets so they didn't escape. And they're vectors of a really important pathogen. And like obviously Chicago is like, you don't like an important like agricultural area. So like if they escaped, then we would have been responsible for this like outbreak of Pierce's. So anyways, so it was very stressful. But so long story short, I'm hoping to get one of these cameras. I'm running a grant right now to get one of these cameras at the Beamline here. So we don't have to do that. That's my main justification. It's like, please, this doesn't work. Okay, next I'm going to talk about a couple of projects that some of my friends, including Denise, who's here, are working on at the Beamline as well, that kind of exemplify these other really amazing facets of insects. So maybe some of you, I'm sure some of you already know this, but that insects hold a number of records for fastest moving organisms. There's some famous examples like chop giants that like close their mandibles thousands of times faster than the blink of an eye. And like things like grasshoppers and fleas that are like using these mechanisms to jump They have these like really amazing spring-loaded latch-mediated mechanisms that store elastic energy by like loading and locking and then releasing it all at once faster than muscle can do alone. So instead of first studying it, the sharpshooters have one of these mechanisms. Can anybody guess what it might be for based on what I've been talking about so far?
Well, I have. Sorry.
It's okay.
Flicking the pee.
Yeah,
exiting.
flaking the pee because they're consuming so much that the fluid wants to stick to itself, like a raindrop on your windshield when it's raining. There's like a size where water is like the adhesive forces overcome gravity, so it wants to stick to itself, and then when it gets too big, it'll like gravity will take over and it'll run off. But these bugs are so small that those adhesive forces are the main kind of driving like component of the fluid over gravity. So they can't produce a jet like we can. So I'm working with a colleague. These are their little butt flickers. This is super high resolution, slow motion video of it. Isn't that fun? And also we made a video with KQED, Deep Look, that my little blue-green sharpshooters are the star. I'm like the stage mom debut. So you guys can check it out here if you want. It's really fun. But there's basically a group of researchers at Georgia Tech that are studying these mechanisms with the colony of bugs that I was raising at UC Berkeley. And my colleague, who's also a part of this group at Georgia Tech, Nami Ha, is studying the difference between adult and juvenile excretion mechanisms. And we used micro-CT both for the blue-green sharpshooters and for this project to understand the anatomy. And then Nami's actually a fluid mechanics engineer, and she's getting her PhD studying this kind of thing, like how these mechanisms evolve over time and how they work in these different creatures. So that's something exciting that she's working on, that we're using micro-CT to like contribute to that. Denise here at UC Davis is using the Beamline to study orchid bees and looking at neurons in the brain. So you guys can correct me if I'm wrong, Santiago is here as well. So I'm nervous again. This is a group of neurons called Kinian cells. They're a center for learning and associative memory. And so Dan is looking at the density of neurons in these areas to see how that impacts visual associative memory. So these little dots here are the neurons of the cells in the brain. So she's looking at like the organization of those, which is just like mind-blowing. So it's very cool.
How big is that, Liz?
How big is this? This is 10x. This is 4x or 10x?
That was first, like, that's 4x.
The 4x.
One
Isn't that
point
crazy?
six.
Yeah, 1.6 micron pixel size. So this whole thing is like, I have it on my computer. Yeah, like a little over, like a couple millimeters wide. This area. I have the parameters saved in like a file on my desktop so I can pull it up. Something that we're also looking at, just very briefly, is my colleague Lynn, who's a graduate student at UC Berkeley, is using 3D imaging to study egg development. And I know spiders aren't insects, okay, but we're gonna talk about them. Anyways, so in different species of spiders that have different mating behaviors, she's studying egg development and maturation, which has been interesting to be a part of as well. So I think these grad students are really are doing really interesting and amazing work in different kind of applications of how we can use 3D imaging to study really different kinds of questions. And I think it's really just the tip of the iceberg in terms of the kind of applications for insects and other life sciences that we could do. There's a lot of people doing really different, interesting, exciting things here, like looking at batteries or looking at rock deformation mechanics, looking at like material science things, like people developing different like types of concrete that are like more structurally sound, less expensive, or looking at membranes for filtering water, that kind of thing. But you know, at the end of the day, I'm a biologist, so I love it when there's more biology proposals that come to the Beamline. So I'm hoping that, you know, this will get the wheels turning and maybe some of you will want to submit an application or talk to me about what that could look like to do some experiments together. There's a lot of people that do batteries, so it'd be fun to do some more creatures. That's all. So that's the first kind of component of my hopes and dreams, is to broaden the biology focus of what we're working on. So that would be really exciting to me and hopefully to all of you as well. I think there's a lot of things kind of like the cryojet that we're using to kind of expand the possibilities of what we could use the 3D imaging to learn about and study. So we have, like, for example, a load cell that like squishes things or stretches things as we're scanning them. We can run batteries like in the Beamline to look at, like, you know how reusable batteries are annoying because they stop working or they get worse and worse over time. They grow like, I don't know that much about it, but they grow like these like dendrite structures inside. So like people look at how those form as they like deplete a battery and recharge it inside batteries, which is pretty interesting. And there's also temperature cells where we can heat things up to like 2000 degrees Celsius, or things like the cryojet where we can image frozen things. And my favorite story, again, it's funny that Andrew's here because basically, Andrew, or this is my understanding of it, maybe you have a different side of it, but basically Andrew was like, well, what, can we scan a tree? And my boss was like, what? Probably not, but like maybe. So they talked to the engineers and they actually dropped us the stage. So they like sawed out this giant piece of like the breadboard that everything sits on and like inserted this motor that like can drop the whole stage. So like this is like a million motors and there's all these wires running to it. Like the level of complexity to like make this happen kind of blows my mind. But there's just like a bunch of engineers that their job is like, when scientists come and they're like, hey, I want to do this. Their job is, and they think it's really fun to like think creatively about like how to do these kinds of like new types of experiments. So this is Andrew's grapevine that on the stage that like got dropped out to scan it. And they've done some really cool, interesting things like scanning live plants and like dehydrating them and rehydrating them, looking at how the vessels change, which has been a really great avenue of research at the Beamline. So yeah, I mean, I also mentioned that I'm trying to get a camera so we can do this kind of thing so I don't have to fly the bugs to Chicago. I think one problem about 3D imaging in general is that it's really hard to get in the door. Like you often need, or you do need like a really meaty computer to do data processing. You need access to a machine. You need help to like learn how to either use the machine or learn how to use the 3D data processing technology. And I know as a grad student, like I got really lucky that when I went into my boss's office and asked him for an expensive computer, he said yes, but there's definitely a world where that wouldn't have been possible. So I think a lot of the things that like me and my role now and like thinking about the future of what this could look like, I want to drop access in a lot of different ways to understanding that. So like, you know, there's a lot of small ways that we're doing that. We're trying to build our computing team, which is, this is the members of some of the members of our computing team are trying to build some web-based tools for people to use to do 3D image data visualization and processing, which is really great. We are, I'm not quite sure how to do this, but like lowering the barrier of entry for like getting access to the facility, which might be a little bit complicated, but that's one of the reasons I'm here, just to kind of share what we're doing so we get more people involved. Also like sample prep. So Denise and I right now, there's a lot of different ways to like stain and fix and scan specimens that there's just like literature is kind of a little bit scattered. So we're putting together a paper that kind of organizes it a little bit better and could provide some recommendations for people that are interested in like preparing specimens for these techniques. Something that I did was, if you guys have 3D image data and you're interested in like making a mesh or making a nice image or something like that, we published a paper, I guess in 2023. So it's not, I can't say last year anymore. That's kind of going through the steps that you would need to take using open source software. Using like, there's like a really straightforward tutorial that has an image of like each step in the process. So you can like actually walk through it. Hopefully you can do it and you can tell me if that's possible or not. The kind of like lower the, like help you get really nice images out of your data. So if any of you are interested in doing 3D imaging at the synchrotron, this, I'd say like kind of this, maybe even the step before all of this is to just chat with me about what you're interested in working on. You can always send me an email or just talk to me after this if there's a question that you're thinking about. And then kind of the next step is to submit a proposal for beam time. But you know, a lot of the times it's helpful to just get a scan for feasibility or something like that, so we can talk about what that might look like too. Or if you're just interested in coming to Berkeley to see the accelerator, it's really, really cool. So you can always come down and get a tour. So yeah, we can even just do something like that if you're interested. I always love showing people around. It's a really amazing place. Something also that we're working on is like a lot of these images are really cool and visually appealing, but at the end of the day, they kind of just go into a repository, which is great. It's great for people to be able to access that, but it's kind of like they go into the repository and then that's kind of it. So I'm thinking about like the big picture of how we can leverage this better for outreach. One thing that I worked on was building a VR experience that takes our 3D imaging data and brings it into schools. So there's like a little, I worked with this company called called Dynamoid that has a program on if anybody of you have an Oculus or a MetaQuest headset, you can just download it for free right now. It's called 10K, like 10K. And you can go on our little adventure that like takes you inside the grapevines using our bug data and some of Andrew and Craig's data of the inside of the xylem vessels as well. It's a little warped in the video because like it's hard to project a VR video on the screen, but it's really fun. And it's gotten better since we made this. So it's, yeah. So I've used it for like education and outreach at like the field day. Have you guys been to the field day? Or yeah, where it's like there's farmers and like people doing like continued education and that kind of thing. Yeah. And in schools to like talk about like plant diseases and whatnot. Yeah. So thanks. Thanks. All right. Go ahead.
I have a quick question. When the sharpshooters are feeding on the phloem, transmit the disease, do they spit back in
Yeah.
when they go to different vines, or is it like their stylet is already just carrying everything?
Yeah. So I think originally the idea was that it's just like a dirty needle, but one of the papers showed that there's a statistical relationship between whether or not it's in that pre-cybarium, so this kind of area with the valve and the flap after the stylet, that's really indicative of if the disease is getting spread or not. So yeah, I think it's something to do with like backwash, for lack of a better word, but there's also like in the xylem vessel, it's pulling on the sharpshooter itself. So there might be something to do with like priming the pump, like regulating the pressure and then taking the fluid out or something like that. So yeah, there's like definitely some interesting questions there that we're kind of exploring with these techniques. So, yeah. Go ahead.
I have two questions.
Sure.
This was super interesting. My first question is, so I think this is a clarifying question. Is the organism dead when you first scan it?
Yeah,
3D?
that's a great question. When everything that I've done in 3D, the organism is dead.
Okay.
The only thing I showed that wasn't dead was when it was
it's the
pumping,
X-ray.
actively pumping, then it was alive. Yeah.
Okay. And is that just like a normal X-ray, or is it like super duper X-ray?
It's super duper X-ray. Yeah.
Okay, so my first question was like, does that the dog? I was just curious, that like messes that bug up.
Yeah.
Or if they're like fine and they're walking around afterwards, they're like totally cool, or if it's like that bug is gone.
Yeah,
I'm just
well,
wondering.
it's it's interesting that it's able to survive.
Okay. You were saying that they're just like spit, like there's so much energy going on.
Yeah.
So my second question is.
But they don't seem to so like it's not good for them. Like there's a lot of protocols that make sure like we don't get in there in the box when the X-ray's on. So like the fact that they are fine for a while is kind of interesting. And there's like, we keep the dosage low, so it's like a lower amount of x-rays. But yeah, it is, they don't see, like, that bug wasn't like attached to the plant. So we put the x-ray on it and it just kept feeding, you know? So it's kind of, I don't know. It's interesting. Yeah.
Okay, so then my second question was, so with that bug, the sharpshooter one, when you were like, I used the 2 x-rays with the video, and then I'd use the snapshots to get like what volume, and then I could like backtrack, back calculate
Yeah.
what volume was being pulled by all my other bugs.
Yeah.
Is that like, you had a few bugs that you did the whole process of the double X-ray and the 3D image. But then you have all these other samples, so like that, can you just explain
I see
that
what you're saying.
process? Like,
Yeah.
how do you
So
actually?
the double x-ray
Backtrack.
is just for like vertebrates, like mice is as small as you can get with a double x-ray. So we were doing a single x-ray video
Mhm.
and then flash freezing the bugs. So I see what you're saying. So maybe I didn't, for the flash freezing of the bugs,
Or like, how do you get your calculations for everyone?
right,
Do you
yeah.
use like a few as your model?
Yeah,
Or,
so
yeah.
we use the 3D snapshots for the model. So basically we flash froze like 40
Mhm.
and they all turned out to be like different, to like get different stages of feeding. And basically, like some of them were empty, some of them were medium, full, like different kind of, we could like string them together like a flip book to get kind of the whole process. And so from there, we can obviously measure the volume because it's 3D, but you're asking about how we then back calculate to the 2D image, right?
And then
So-
like, is each individual is at a different point of feeding?
Yeah.
So it's like
Yes.
you're understanding.
stringing
that
them
they're
together
all
to-
different, so it's okay though.
Yeah.
So then you're gonna take no, which is fine, I'm just
All right,
thinking
reviewer
about like
too.
other
I'm just
systems.
kidding. That's a great question.
So then like after you make the curve,
Yeah.
like you have everything.
Yeah.
So then what do you do if you want to calculate for others? Like now I'm with you.
Yeah,
Okay.
no, that's a great question. And there is like some disparities between individuals,
Mhm.
right? And that's an important thing to think of as well. So yeah, so basically we took the volume, yeah, so there's 40 different bugs. Each one of them is showing like a snapshot of a different part of feeding. So it's not one bug showing the whole thing. So there is error in there for sure. It's better than anything else that we have. So
That is, yeah, I'm just.
but okay, so we have the 3D, and then we found by chance that there's a relationship between if you take the 3D image and you like put it in the same side that you're getting the 2D image,
Okay.
and you like measure the largest distance from the top and the bottom, then there's a relationship between that, like a linear relationship between that number and the volume. So we just plotted like that number, the
Okay.
2D number and the volume, and found a linear relationship. So then we went back to the images and we like found the formula of that line, right? So then we could go back to the 2D images and like measure that line and use it to back calculate the volume.
Okay, yeah, that's making sense. I'm just thinking about other systems while you're
Yeah,
explaining this.
I
So
don't
I'm trying
know.
to think about like.
Like how you would do that, because that's an
like,
important
I'm
part of
thinking
it.
about other systems
Yeah.
right now, so thank you for.
100%.
explaining this.
Yeah, because you're right. Like you're absolutely right.
Yeah.
Sorry for calling you a reviewer too. I was just making a joke.
No, I'll.
But you're absolutely right. Like that is a particular relationship that we got lucky for in the system. And with XROM, like it's pretty straightforward. Like it's a pretty straightforward mechanism. But like for us, because we only have the one X-ray, we have to kind of get lucky with something like that. So like I'm not totally sure. I think it would be really cool to like look at, like you have to be confident that like the measurement that you're getting, you can connect to the 3D system. And maybe there's systems where that wouldn't be that necessary. Like if you're not looking at volume, for example, like you might not have to do that. Like maybe there's like, if we're looking at like how insects like jump and store elastic energy, maybe there's like a parameter for like, I don't know, estimating like the amount of like deformation that you're going through before. Yeah, so I'm curious once you cook once that cooks a little bit, what you come up with. So yeah, what do you study?
I was thinking more in terms of like, so right now I'm having a challenge with in chemistry of capturing sulfonated compounds. So it's like adjacently related to this,
Yeah.
but using like click chemistry, so you can like derivatize compounds to try to capture them. And then trying to use click chemistry to figure out where these compounds go within an organism. So I'm just like
Yeah.
connecting some dots
Okay.
on how like 3D, how you can like connect multiple tools together
Yeah,
and like what's frozen, what's still versus what's moving
Yeah.
versus what's not, because in click chemistry, that's like moving.
Yeah.
So, but this is still, so I'm thinking about how you can like integrate
Yeah.
to, yeah. 'Cause,
well,
like,
cool. Well, I would be curious to
Yeah.
hear more about that. And there's there's other Beamlines that do like more chemistry focused. So yeah, we can talk about it. That would be fun. Yeah, go ahead.
I have another quick question for
Oh,
the
sorry.
plants that are diseased.
Sorry.
For the plants that are diseased, do their like xylem tubes, are they altered? Like, do the bugs preferentially feed on the diseased plants? Are they young leaves? And like, how does that?
Yeah, yeah. I don't know. Somebody knows if there's a preference. It does block the vessels, so eventually they'll get so clogged that they won't be able to, it's not transporting fluid anymore, or there's cavitation, or the pressure's too high. I'm not sure how the infection, it would change like the width of the vessel, and I guess if they have a preference for larger vessels, maybe, yeah, maybe it would affect it. But that's an Andrew question, and he already left, so.
I didn't know. CT scan the diseased plants too.
Yeah. Sorry, go ahead.
I was curious. I was curious for the fluid mechanics model. I was curious, like, you said you played around with like the angle of the input of the
Yeah.
little needly part. And I was curious if you did like a general like sensitivity analysis
Yeah.
of the computational model of just, I don't know, either like the rate at which the liquid's intaken, or like even like from the perspective of the plant, like the ability to inject the needle or not be able to for like disease.
Oh, like
transmission.
so there's two things there, the sensitivity analysis, and then like if we actually looked at like, yeah, I think the beginning of this process is a really interesting juncture that is going to be complicated to model if you're a fluid modeler. I hope. Really cool.
A modeler, but not a
Okay.
fluid.
I'm like, if you could help me. Because you're right, because you're looking at a system that's under tension and then you're inserting a needle. Yeah, like that juncture is really interesting, for sure. And it's something that I'm not experienced enough as a fluid modeler to be able to do. My spouse is an aerospace engineer, but I can't afford them to work on this. And then the second part is the sensitivity. So like we did like a mesh sensitivity analysis. I'm not sure if that's what you're talking about, but basically we looked at like different like arrangements of the triangles that like make up the tubes, and to see how like, so that's where we got stuck with the angle of penetration, because you might be a reviewer too. Because we found that once we varied the parameters like within the mesh, then it kind of fell out and fell more closely into like the error range. So yeah, that's something that we did find, but the other ones seem to be robust. So that was kind of interesting. And there's like, within that, there's plenty of things that you could play around with. Like you could, yeah, there's like a lot of different, so we had to kind of like narrow the directions that we went in. And we picked the things that we did. I don't know if there's something else that would be more interesting to look at, but it's the model's there, so we can play around with whatever we want. So, yeah, go ahead.
I have what's probably gonna be a much easier question than all of these, which is just something that I always wonder when I see these. How are they colored? Like,
Oh,
are
great.
you manually coloring
Yes.
them?
Yes.
And
Yes.
so that's like a
large
you
part of
must
my job
have
is coloring.
biological expertise in this thing to have some understanding what these different tissues.
Yes.
Because like when I look at the gray version, sometimes I'm like, I don't even see the difference between the two colors, and
Yeah. Oh, yeah. 100%. Yes. Yes. So like people always talk about like segmenting and like, oh, you can get an undergrad or whatever, but like you need to be an expert on this in order to do it. And so like for me, this is something that I enjoy. I like put on reality TV and I like just color. I'm like, I have to do some coloring. So like you see the structures based on the density. So I'm not like drawing random shapes or whatever. Like you're getting a 2D slice and you're looking at like the different pieces of anatomy that are like picked up based on the grayscale value. That's determined by the density.
Okay, so this is a composite of you doing this on a shiz ton of 2D.
Yes. And then it's colored because I brought it into another, I use, this is the program that like people who aren't at Disney that do 3D animations use because they have their own proprietary software, but this is called Autodesk Maya. So like the Avengers, so like the CGI and like Avengers or, what's that movie with Ryan Reynolds where he's in the desert? Blade Runner, whatever, is that it? I don't know, what is it? Anyway, so like any movie that's not Disney that uses CGI probably uses this software, like Monsters Inc., for example, it uses the same kind of modeling software. So I pull so basically I take my 3D image data, I like color code the different regions of the scan. I just break them into like maybe 10 different, each one of these colors is like a different region. And then I can pull them into this other software to take like an image where I like kind of smooth it out and make this transparent or whatever. So yeah, there's a little bit of a system. That's a great question. Because sometimes it seems like did you just make all that up? But yeah.
Thank you for
For
clarifying.
sure, yeah. I should maybe make a video. It would be interesting to make a little video of like the process to kind of inform. It's hard when you don't know and like sometimes you see it in a book that has this simple schematic and then you look at the 3D image and it's like, what the heck? Like it's so much more complicated than that, which is why I love doing this because it's so cool to like look under the hood and be like, wow, this is not what this drawing says. Like it's so much more complex. So to me, that's really exciting, but it does make it really difficult. So I've watched a lot of reality television. So for better or worse.
Thank you. Cool.
If anyone is interested in talking